Yeah, good evening, everyone. Welcome to this live session for the course Human Molecular Genetics. So we have suggested that all those who wish to have few queries discussed, you can add these queries on the Google form. There are some 5-6 questions that have been posted, so we will take each one of them and then discuss. And those who have joined the live YouTube session, you may also post your queries using the chat box that is on the right side of the YouTube window. So there may be some lag period for us to see the queries that you post in that live chat box. Therefore, you please wait for your term to come. So I'll try to answer queries that are relevant to this topic. So if there are questions on topics that have already been covered in the lectures, I would refer that you please go and look at the lectures again. But there are some clarification needed or more generic questions we'll take up. Yeah, so with regard to questions on whether you can have a lab training, internship, and so on, I would suggest that you please write to me directly over email, because that is not something related to this course in indirect way. So I don't want to waste time in answering those questions for those who are not interested in that topic. Okay. So you can always write, on that, either in discussion forum or directly you can write to me. So welcome once again, all of you, for this course. Good evening to all of you. So many of you have joined. I see that about 70 plus students have already joined in this live streaming session. So it would be a good session, I guess, for you to learn something may not have been discussed right here. So let me take up the questions that have already been posted in the Google form. Then we'll go to the questions that are there in the YouTube chat box. There is a question I want to know about some research related, such as how to write a research paper in the field of biotechnology. I think there is a course concurrently offered, of course, one by me, but there are also other faculty who are offering on how to write research papers, projects, scientific communication. So it's a very detailed, topic, cannot be explained in 2-3 minutes. So I would suggest that those who are interested in such kind of topic may enroll for that program. If you are not interested in enrolling, you may want to look at the YouTube, lectures that are available freely. Some of them are my own lectures. The other one is that on internship, whether you would get that is something that I would suggest that you please off the record you write to us directly. I don't want to take up that kind of queries in the live session. This is for the topics that are covered in the lectures for this particular course. There's one question about epigenetics. How does methylation affect the characteristic changes of individual? Yeah, obviously, the methylation is a change that happens in the genome. As a result, how the genome functions in terms of its expression of the genes and so on, that is regulated by, to some extent, the methylation of the genome. So epigenetics is nothing but the changes that one of the modifications is methylation. So it certainly would either activate or inactivate a gene or change its expression profile. So that certainly would have an impact on the phenotype. It could be anything from susceptibility to a disease or to behavioral changes, right? So this is being studied well. So that is how it acts on. So it acts through the gene expression patterns. So that's being well documented now. So let's go to some of these questions that are there in the live chat box. So someone is asking, what is the general scope of this course? I mean, this is a course that helps us to understand how we study or understand changes in the gene, in the human genome, affects the individual in terms of being a normal person or when you have the disease, right? So what are the approaches you take? How do you really study? How do you find the gene? So that's the scope. So practical implication, I mean, obviously from diagnosis to treatment, right? So these are directly related to many of the disorders. So it's something as a direct impact on health, right? So that's, if you know a gene mutation therein results in a disease. So, and if it is inherited in a family, then you can do a prenatal diagnosis to test whether the individual who is carrying a child, whether the child would have the disease or not. So that has an immediate relevance to the family. So for that, you need to know what is the gene and what is the mutation. So that is as simple as that. To treatment of genetic disorders, like if you know that this is the gene that is defective, and what are the pathways that are affected by the mutation in the gene. Then if we can understand that, then we may be able to rectify or restore the pathway, and that is leading to the therapy, right? So these are, it's exactly falling into human health. So that's the scope of that, implications. Kindly explain the construction of genes in RFLP using questions in practice session 2. I don't know the practice session 2. I think this is handled by one of the TAs. These are questions that are taken from the old question papers or assignments, I guess. So I don't know what that TA has done. So I would recommend that you please write to the TA once again, and so we'll be able to do that. But anyway, so that's the we can do that. But I cannot use here chalkboard and other kind of a thing, so I cannot explain. So to give you an example, the RFLP is nothing but restriction fragment length polymorphism, right? So you have a segment of the genome. There you have, 1 or 2, 3 sites for a restriction site, restriction endonuclease. Now, if there are mutations or changes in the sites for the restriction enzyme, then the enzyme may not cut there, right? So as a result, there will be a difference in the size of the fragment that are being made. Now that, if that is linked to a disease-causing gene, then that helps us to track, the segregation of the gene. That's the concept. Maybe I would suggest that you please put a particular query in the discussion forum, and the TA would again explain you the whole process that has been explained in the lecture. Okay, I would urge you to do that. It will be explained. Yeah. So what further we can modify in this branch? I really do not understand the question. What is the latest modification made in this branch? If your question is referring to what are the new approaches, techniques, and inventions, whatever in this field, that perhaps you may be trying to ask that. But yeah, that is approaches, but the concept remains the same. In this course, we have discussed about how we can look at the so-called monogenetic disorders, and then how you look at the polygenic disorders. Then how do you identify genes in both of these conditions? And that's the traditional way of understanding. But what people may do now because of the technology evolved in terms of sequencing, and sequencing has become much more cheaper, inexpensive, and quicker. People do, instead of doing uh, a looking at a candidate region, they may sequence all the known gene sequence in the genome, and then try to ask whether any of this mutated. But it comes with its own challenge, because there are, in a given individual, there could be 50 different genes that have variations. So which variation is indeed causing the disease, and which variation is affecting the the physiology, is very difficult to understand, unless the mutation that you see are like large deletions or frame shift mutations. So this is not likely that in every family that you study, you would find that kind of a mutation. So it only are indicative that people use, identify some 20 different genes. But then if it is a given gene is not known for a given disorder, then such kind of studies really doesn't give you any conclusive proof as to which is the gene that is defective. So one has to go back to the traditional method and looking at the co-segregation of the gene variant in that family, and see whether that variant pass along with the phenotype, and then do some functional studies to understand. So that's pretty much, again, you have to come back to the drawing board. But if the gene is known for a given disease, then sequencing becomes much easier. Yeah. So that's the difference. We have a newer sequencing technologies, and these approaches are also discussed in another course called functional genomics, which may be offered in the next semester. So those who are interested may join that course, or these lectures are available in the YouTube, so you can go through them. Yeah. What is the relationship between genetic screening and anthropology? These are anthropology is something you look at. You can say that human evolution. It's not necessarily based on the DNA. It even based on the language that you use, the tradition that you do, your customs and the way you worship or whatever. That is anthropology. But genetic screening is purely looking at genes. So it's, you can use genetic approach to strengthen an anthropology finding, but anthropology is not that way genetics. Okay. Autosomal dominant. Why do we always assume the affected to be capital A, small a? But if the concept penetrance generation could be between otherwise. Right. I mean, so there are, what you say is that even if one of the two, we are talking about autosomal, so even if one of the two copies is mutated, so you have only one copy of the mutated gene, then you would have the phenotype. So that's what dominant. That's why it is capital A, small a. But the penetrance is whether or not you would show phenotype. At times this is modified by something else. So you may have a genotype for the dominant disorder, but you may not show the phenotype. But all those individuals who show phenotype will have one affected allele. Therefore, that is still correct, right? So that's what it is. So it's not that every dominant disorder show penetrance issue, right? There are many which are 100% penetrant. There are some that could be 70%, 80%, depending on which gene and which phenotype you are looking at. So, but invariably that affected individual will be heterozygous for the affected gene, meaning only one copy being affected. So in that way, still it is valid. What are the different processes we can use to separate DNA, double-stranded DNA into single-stranded DNA? Well, I mean, there are processes people use, columns, affinity columns, but I don't know. These are all old methods, but nowadays nobody use them for separating. But you need to understand that if you have separated the two strands and leave it for some time, they are going to anneal back, right? these are kinetics people do, like, you separate them and look at fraction of strands that remain as single-stranded form as a function of time to understand the sequence complexity. That's what we will do. And that they use what is called as hydroxyapatite columns to pass through and find out how many of them are still single-stranded, how many of them are double-stranded, and so on. So it basically looks at the affinity, right? So you can read more on that. That's one method here. Facts are facts, sir. Whether assignment topics are enough to cover the whole topic. Facts are fact is that no, it is not. So assignment is going to test part of the topics that are covered. So there are some topics that are not covered by assignment, but it tells you how much prepared you are, right? That's what it is. Yeah. How can you find the major and minor grooves in DNA? Well, these are it's not something that is related to this course, but that is more on the structure of the DNA. So the people do it at atomic resolution, right? You don't deal with in this course, but then that is how it is. You can go back and study. I mean, I don't want to discuss on this in this course. Yeah. There is something says that volume is low. I may suggest that you increase the volume in your whichever cell phone or use earphone or something. That's, yeah, maybe I'll try come close and then say, yeah. Murali has, Mr. Murali Krishnan already responded saying that, there's no problem with the audio. Maybe there is a problem with your handset, okay? Yeah, there's a follow-up question that I was asking about how to separate two strands of DNA. I mean, these are not easy to do. It's impossible. There's no way we can handle or hold two strands and then pull them apart, right? It is so small that there's nothing that you can really hold and separate. So that is technically not possible as of now. Yeah. People, try to do something like they try to fix one end of the DNA and allow it to reanneal or separate, but that is doable, right? But something like fork, you can hold it and pull it, is right now it's not possible. Yeah. What are some good topics to write review paper based on human molecular genetics? Right. I mean, this depends on your interest. There are so many topics that are covered. What topic that interests you? You want to read more and write, right? This is about 10-hour course. So, in every 1 hour lecture, at least there are 3 topics that you can take up for a review, right? It depends on what is your interest. Can we merge human genes with other species genes? I really do not understand what you're asking. Are you asking that can we use the human genes and put in some other animal? That is normally done. Like most of the animal models that you see, transgenic animal models, that is what done, right? So you put the human gene there and then let it express and then develop. If it develops the phenotype, disease phenotype, that is what looked at. So that is doable, technically doable. I don't see any issue there. Yeah. Haplogroup. I am not too sure what you are asking haplogroup, or are you asking about haplotype? Because we discussed haplotype in our in the pedigree analysis or whatever. That is the various alleles of markers that are physically linked. They get transmitted as a block, so that is called as haplotype. For example, marker 1, 2, 3, 4, 5. They are present in a given segment of a genome, or a chromosome in that order, then the alleles of that markers would invariably segregating together as a block, because there may not be any recombination within that. So that's what we call as haplotype. Yeah. Yeah, someone is asking this question about it's Harshavardhani, I guess, again on the penetrance problem. That yes, I mean there could be an individual who is uh, having a genotype for the disease but not having the phenotype, like carrier, so to say, because of the penetrance issue. Yes, that is possible. Um, if you know the gene, then one can do sequencing and then tell exactly that this individual is having the disease allele, not showing the phenotype. But if it is if you don't know the gene but you are looking at the pedigree, then you can only calculate as to how many minimum number of individuals that are having this are likely to have the affected allele yet not showing the phenotype. For example, an individual in the third generation, say suppose, is showing a phenotype, and in the second generation, his father is asymptomatic. Therefore, you know that, but in the first generation, that is this third generation guy's grandparent is having the disease, that means that that phenotype has skipped one generation. So that parent should be, one of them should be a carrier. So you calculate, the penetrance based on that kind of a prediction. That is how you do it. Yeah. And it clarifies cDNA specifically on DNA sequencing. I don't know what exactly again the question is. If you are talking about how do you sequence cDNA, see, cDNA is copied DNA, meaning the mRNA is used as a template, and then you make a new strand which is copying the mRNA. And then this DNA is again copied to make it double-stranded DNA, and then you clone it. Then it eventually it is a double-strand DNA in a vector. So you sequence just like any other DNA sequencing that you would do. So there is no difference in how do you sequence cDNA. So there is a question called as what are silent mutations versus variation. Variation is a very generic term. Any change in the DNA is called as variation. A variation could be a mutation if it affects the gene function, or it is present, some change is present in the region that is not corresponding to any gene or gene elements, then it may not affect any of the gene function. So therefore variation is a very, very loose term to tell about very changes in the DNA, and more often it is referred as something which doesn't affect the function of the genome. But if you talk about silent mutation, these are you have a base change in the coding sequence which does not change the amino acid it codes. So it's still you have a change DNA, but again you have the same amino acid coded by that. That's often referred to as silent mutations. Okay. Which book would you recommend based on genetics? I don't know for which course you're asking, but it's human molecular genetics. There is a book that is recommended, right? That name of the book also is Human Molecular Genetics. But we have, provided many reading material as part of this course. So if you read those topics that are given, it should be good enough. Even this transcript of this course, the lectures are given, so that's fairly good enough for you to read. Why salt is important DNA precipitation? Anyway, so this, yeah, how do you precipitate DNA? You have to, you add alcohol to get the, water out of the DNA. Therefore, that what DNA is not in dissolved form now. It forms the precipitate. So that's what that process. The salt helps in that process. That's what it is. How is denaturation of the DNA is related to temperature? Yeah, I mean, you can do denaturation multiple ways. One way is temperature. You have to break the hydrogen bonds, so you can use some, sodium hydroxide too. So there's a chemical reaction, right? In that sense. Why blunt end cloning generates 50% sense, 50% antisense clones? Well, I mean, this is, I think you are referring to some coding sequence of the gene. So if when you do a blunt end ligation, a fragment can get ligated this way or this way. So there is a probability that 50% go this way or that way. So that's what you call as sense and antisense, I presume. Difference between gene silencing and repression. I don't know in which way you are saying this. Gene silencing is a method that you use to silence a gene, right? One way people do it is RNAi is also called as gene silencing. So you basically remove the RNA made by a given gene, therefore it is equivalent to not having the gene. Repression could be even at the level of transcriptional control, right? You can have some blocker chemicals or whatever it is which blocks the expression of the gene, if you know the transcription factor. That's the loose term. I don't know exactly what you are asking. Right? Yeah. How to solve a pedigree analysis in a very, very easy way. Very simple. Understand the concept, you can solve. So there's nothing, there is no shortcut. Right? You understand, it's very simple, right? You have male, female affected marriage. This is, that's all you have. Now you have to, whatever problem that is given to you is that monogenic problem, that is a given gene is affected and it is a dominant or recessive. So if you apply the concepts of Mendelian genetics, you should be able to answer any question. So the easy way to understand the concept, and if you know the concept, it's easy to solve any pedigree related problem. Yeah. I guess we have covered pretty much all the questions. Maybe I'll wait for a couple of more minutes. So if there are more questions, then we will take it up. If not, then I would recommend you continue going through these lectures. If you are unable to follow them, go back and listen to the lectures once again. And if you still have any specific questions on the topic discussed, if you are not following something or something, you can write in the discussion forum. We'll try to help you out. Somebody is asking overlapping fragments. That's all written, but if the question is what is overlapping fragment, the answer is there. There are two fragments. They share some sequence between them. So that is overlapping fragments, right? So, and they are very, very important for you to build the entire genome, because only with the overlap you know that they are close to each other. Say suppose this is the fragment, your cut in between these two became two different fragments. So you don't know whether they were like this or like this. They are from different chromosomes. You don't know. So the only way you can anchor two different fragments together is if they are overlap, right? So that's very, very important. If that's the question, that's the answer. Great. I think we have covered most of the topics that has been asked here and relevant to the topics that we covered in this course. And, I recommend all of you to go through the course lectures, read the reading materials, and then do well in your studies, because I'm sure many of you are doing it as part of your credits to your program. So you can always study well and then get a good grades. That's also important. But and then of course this topic is something that is relevant to your everyday of life. If you look at your family, there may be someone who may have a genetic disorder. So this understanding helps you, in even handling the disease and so on. So that way it is far more relevant to the society. So I'm sure all of you have appreciated that particular aspect of the course and found it useful. So if you have more questions, hold on. So we would in 2 weeks from now, we'll have another live session. So we'll take up again questions if you have any. So we can do that. Yeah. Good. And that's all for today. We'll see you 2 weeks from now. All the best for your studies, even other courses. Yeah. Yeah, Murali, we could end the session now. Okay?
Thank you so much, sir, for your valuable time. We will stop the broadcast now, sir.
Yeah, thank you. Thanks a lot. Yeah.
Thank you, sir.
No.